Hello everyone, and welcome to my YouTube channel. I'm here to discuss replication of DNA viruses. The genomes of papovavirus, adenovirus, and herpes virus are transcribed and replicated in the nucleus and therefore can utilize transcriptional enzymes of the host for messenger RNA generation. Most DNA viruses contain a single genome of linear double-stranded DNA. The papovaviruses comprising the polyoma and papilloma viruses, however, have circular DNA genomes. The transcriptional program consists of at least 2 cycles of transcription for papovaviruses and at least 3 for herpes viruses. In each instance, the structure of viral polypeptides are made from messenger RNA generated from the last cycle of transcription. There are several steps in replication of DNA viruses. This includes the first step is attachment. Attachment involves specific binding of a viral protein, usually referred to as the anti-receptor, to a constituent of the cell surface, which is the receptor. Complex viruses, such as herpes simplex virus, HSV, may have more than one species of anti-receptor molecule. The cellular receptors identified are largely or mainly glycoproteins, but may include salicylic acid or heparan sulfate. Ions in concentrations sufficient to reduce electrostatic repulsion are required for attachment, but it's mainly temperature and energy dependent. The second step in the replication of DNA viruses is penetration or entry. Penetration is an energy-dependent step, and it occurs almost instantly after attachment and involves one of the 3 mechanisms: endocytosis of the virus particle resulting in accumulation of virions inside cytoplasmic vacuoles, translocation of the virion across the plasma membrane, or fission of the cellular membrane with the virion envelope. Fission of the viral envelopes with the plasma membranes requires the interaction of specific viral proteins in the viral envelope with proteins in the cellular membrane. The third step in the replication of DNA viruses is uncoating. Since they are enveloped, it involves the removal of this envelope, which is uncoating. Uncoating is a general term applied to the events occurring after penetration, which set the stage for the viral genome to express its functions. Herpes virus, adenovirus, and papillomavirus nucleocapsids are transported to the nuclear pore, where the viral DNA is released directly into the nucleus. The pox virus genome is uncoated in 2 stages, whereas in the first stage, the outer covering is removed by host enzymes, leading to the second one is the release of viral DNA from the core, which appears to require the participation of viral gene products made after infection. The fourth step, replication of DNA viruses, replication or multiplication of the genome. All known DNA viruses infecting vertebrate hosts contain monopeptide genome, except for the papova virus genome. All are fully or at least partially double-stranded. Although pox virus DNAs have been detected in the nucleus, the transcription and most of the other events in the replicative cycle appear to take place in the cytoplasm. The genome is transcribed by a viral enzyme. The initial transcription occurs in the core of the virus. Here is a flow of events during the replication of the herpes viruses. The parental DNA is transcribed into messenger RNA, and which is now translated to proteins. This occurs in two double or two circles. Then the progeny DNA is transcribed from parental DNA, and this is included in the progeny virus. Still on replication of or multiplication of DNA viruses, one human parvovirus, the adeno-associated virus, known as helper viruses for the multiplication. In the absence of a helper virus, the genome appears to integrate into a specific locus of a human chromosome. Other human parvoviruses are capable of multiplying without the assistance of helper viruses. Viral replication involves the synthesis of a DNA strand complementary to the single-stranded genomic DNA in the nucleus and the transcription of the genome. The DNA of DNA virus, for example, hepatitis B virus, is first repaired and converted into a closed circular molecule by a DNA polymerase. It is packaged in the viral and transcribed into two classes of RNA molecules. That is, a messenger RNA specifying proteins and a genomic RNA, which is transcribed by a reverse transcriptase to make the genomic DNA. Here is a summary of the flow of events during the replication of the hepDNA viruses. For example, hepatitis B virus. There is the genomic circular DNA, which is repaired by viral enzymes into a closed circular DNA, which is transcribed into a positive strand messenger RNA, and then this is translated into proteins. The fifth step is translation. The messenger RNAs are translated with the production of newly synthesized proteins, the capsomes, and the viral proteins are coupled as viral protein coats. That is, the capsomes are assembled around the genome as capsid or protein coat. The next step is assembly and maturation. The herpes virus nucleocapsid is assembled in the nucleus. Unlike other envelope viruses, the envelopment and maturation occur at the inner lamina of the nuclear membrane. The envelope virus accumulates in the space between the inner and outer lamella of the nuclear membrane in the cisternae of the cytoplasmic reticulum and in vesicles carrying the virus to the cell surface. The envelope virus is uniquely shielded from contact with the cytoplasm. Herpes viruses are cytolytic and invariably destroy the cells in which they multiply. Like other envelope viruses, herpes viruses impart to the infected cell new antigenic specificity. The last step in the replication of DNA viruses is the release of viral particles. Envelope viruses are released by bonding from the plasma cell membrane of a host cell. Here are selected references for further reading. Thank you for listening.